In exercise 2.4, we will do histograms of the p-values. So again, we will use the prepared volcano data set. And we want to do histograms of the original and the adjusted p-values. And we will play around a bit with the bins and bin width arguments. We will also find out how we can manually define the borders of the bins, which are called breaks. And yeah, use that to create a histogram where the bin's length is 0.05, but it starts at 0. Okay, so we read in the data set. And first we will do a histogram of the original p-values. So the mapping is quite simple. We just need an x. This is the ANOVA p-value. And we use the geom histogram function. This looks like this here. We can do the same with the adjusted p-values. So we use a different column here. And we can see the shape is quite similar, but you see here that the number of low p-values here in the first bar has dropped. This is normal for adjusted p-values because you make the p-values larger, less significant, but more reliable in this case. So we play around a bit with the number of bins and the width of the bins. So for example, here we say we want to have 10 bins. You can see that these bins are getting larger and you may lose a bit of information. But on the other hand, if you make the bins very small here, like a really low bin width, yeah, it basically gets way too detailed and is also not maybe what we want. So it may make sense to be a bit careful what you choose. And if you want to manually define the breaks, you can use the breaks function. And if you go back to here, one of the larger bins, you see that the bin doesn't start at 0, but actually below 0. But below 0, we can't have any data points because p-values are always between 0 and 1. Also here we have the last bin going over 1. So we want to make the bins a bit more fitting to our situation here with the p-values. And a bin width of 0.05 seems to be quite reasonable. So, for example, the first bin will contain all the significant proteins between 0 and 0.05. So here we actually define the sequence. And you can see here, these are the breaks that we want to use from 0 to 1 and 0.05 steps. And if we do that, you will see here that the bin starts exactly at 0, it ends at 1, and we have bins that are 0.05 wide. In exercise 2.5, we are doing a density plot. So here we will use the long format dataset containing the clinical data. We create a density plot of the log2 transformed intensities. We color them by group, or we color the area by group, and we set a lower transparency or alpha level. Okay, so we read in the data here. Takes a minute. And then we use here the geom density function with reduced alpha of 0.3, for example. And we use for the x-axis the log2 intensity. For the fill, which is the area of this the density plots, we use the group. And here we can see the results. So this is lower density plots over all samples and all proteins in the two groups. And we can see some minor differences between the two groups here by the red and blue colors. In exercise 2.6, we will have a look at box plots of the whole data set. So we again use the long data format with the clinical data. We have box plots for each sample, like we already did in exercise 1.3. We use the group column for filling out the color. You will see what is the difference to the resulting plot in exercise 1.3, where we use group for the color aesthetic. Then we also change the code to create violin plots and combinations of violin and box plots. I've already read in the data here. We use the box plot here with the fill aesthetic for group. And what we see here, that if you use fill, then the inside of the box plot is filled with a specific color and not the lines and the outliers and so on. So this is the difference between fill and color. If we want to do violin plots, the easiest way is to simply swap the geom box plot with the geom violin function. So we can have violin plots which show a bit more the density distribution of the different samples. And we can combine them by simply adding both geom functions. And then we have here a combination. So basically violin plots which have an overlaid box plot here. For exercise 2.7, we again do some box plots, but this time single proteins. So we take the long format data set, filter it only for one specific protein, and then we create box plots that compare the log2 intensities of this protein between HCC and control samples, so between the two groups. And then, additional to the box plots, we also add the individual data points using the geom jitter function. So we read in the data. We then filter the data for the specific accession. And then can use the geom box plot with the following aesthetics. So on the x-axis now we don't have like the name of the samples, but the group. On the y-axis we have the log2 intensity, and we again fill it by group. We have the geom box plot, and we have the geom jitter, which adds the individual data points with a bit reduced transparency value, and a width that corresponds to the width here of the box plot, so that basically the range of the jitter is roughly the same as the width of the box plots. I will show it in a minute. Here also we have a seed, a random seed that we set, because the jitter is a bit random. So we have the seed to always get the same plot basically. So here we can see the results. So we have these two box plots here. One is for the control group, one for the HCC group. All the data points belong to the same protein, and you see that the proteins are, and you see that the individual data points are plotted here, and the jitter basically means that they are randomly distributed over a certain width around the box plots. So that this data point here is here, for example, and not somewhere here or here, is random. But it's jittered a bit so that they are not all on the same line or something